it's a great pleasure to come along. I always enjoy these sessions, and it's fantastic to see the room so full. It's fantastic that there's that amount of passion for the things that we love and care about and are studying. So Maria's given me an excellent background. In fact, I think she looked through my slides and decided she'd try to explain a few of them beforehand so that they made sense to you all. So what I wanted to talk to you about today was a history lesson. Actually, not a history lesson, a sort of a timeline that goes from, that encapsulates how our ideas and understanding of cancer has evolved over 2,000-3,000 years, and bring you right up to the front. I'm going to finish off with unpublished data that shows you where we want to be right now and how we are beginning to turn that knowledge into things that can save lives and have in fact saved some lives. I'll tell you that story. That's not published. That's encapsulated in these slides here. So maybe if I go on. So I think of cancer as evolving in three distinct phases. The first phase is cancer as an anatomic disease, and that's where we define a cancer by the organ in which it arises, in this case a breast cancer on this mammogram, and that's turned out to be incredibly useful as people have understood anatomy better through the work of people like Vesalius, John Hunter, and this fellow down here is a famous American surgeon called William Halsted. And William Halsted took advantage of the anatomic concept of cancer to cure women with breast cancer through the mastectomy procedure which he developed. So what that tells you is anatomy is useful because if we can get a cancer early enough, we can remove it surgically before it has spread. And it's mostly spread of cancers that ultimately claims the life of those who are affected by it. So surgery is critical to cure, and particularly for the diseases I treat, which affect a young population. Without surgery, I don't cure anybody. So that's true to this day. I've actually mistakenly, in effect, implied that the anatomic era of cancer has stopped in 1838, but that's not actually true. It continues to be important today. What happened in 1838 is that this fellow, Rudolf Virchow, a German pathologist, took advantage of a new technology, the microscope, to look down the microscope and suddenly realize that these lumps of flesh were actually composed of cells. And he did this description of what he was seeing down the microscope back in the 19th century. And that led to an incredibly sophisticated classification system that was layered on top of anatomy to describe cancer on the basis of its appearance. And they stained these things, so that originally they were black and white, but they stained them with colors like this pink and the blue, hematoxylin and eosin, which we still use in the labs today. And a pathologist would look at that and say, aha, that is not just any cancer arising in the breast, that is a dedifferentiated leiomyosarcoma, a cancer of smooth muscle. And we still use this classification today. For example, if that were a leiomyosarcoma, I would tell my patient, we should give you radiotherapy and then remove the tumor to try and reduce the risk of the cancer coming back in the local site. But if it was a Ewing sarcoma, another sarcoma that I treat, I would know that I'd need to give that person chemotherapy, because by using chemotherapy, I could triple the survival of that patient. So the light microscope gave us patterns that we'd recognize, that we'd associate with the benefit of certain treatments. It was all done empirically. Thousands of patients were put through these treatments to work out what worked and what didn't. There was no reason behind it, as it were, but it was something that we found incredibly useful over the subsequent 170-odd years, and we still use it today. And then the third era came in, and that's the molecular era. And this is important for a number of reasons. This is a picture of as you see here, James Watson and Francis Crick in 1953 posing for the media in front of a model they built out of a retort stand, and an old laboratory retort stand, describing their vision for the way in which the double helix formed, and that was represented physically here, and Marie gave you a very nice description of it. That was the first time that anybody got that, and it's an epic moment. It causes goosebumps on my skin thinking about how important that contribution was to our understanding of cancer. Because cancer, as I'll show you, is fundamentally a disease of genes. In fact, life is a genetic condition. You've seen this picture, I hope, on the round. It's a beautiful illustration of this point. There are approximately 70 trillion cells in the average human being. each of which arose from one cell through hundreds and thousands, billions of cell divisions to create an average adult. And each cell contains DNA, and this is what's called multicolor fish, which stains each chromosome differently. So this is just the complement of one cell, and that DNA packages up into this linear sequence of letters, which can be considered like the letters in a sentence that conveys meaning. And every aspect of human biology, every aspect of any animal biology, any cellular biology is driven by what's coded as Marie nicely outlined. And that is, unfortunately, also true of cancer. So this is also one of those fundamental breakthrough moments, a paper written by a guy called Peter Noel in 1976, which illustrated the concept of cancer that we currently think of today. So as Marie described, you start off with a normal cell, well, whatever normal means as we get older. It drifts and mutates a little bit, all the cells drifting and mutating. But eventually it gets that critical point where it acquires the switch, it has that event, that change in the sequence, which trips it from being a normal cell into one that starts dividing uncontrollably and ultimately spreading, and all those other properties of cancer. And you can see this expansion. Peter Noel described the subsequent acquisition of second events and third events, and some cancers have thousands of events which have occurred because of the nature of they lose control of their genomes, in effect. And this was described in chronic granulocytic lymphoma, leukemia, or chronic myeloid leukemia. And Peter Noel was famous for this. He formulated this hypothesis in 1976. But in fact, he was the first person to see a human mutation in cancer. He published a paper in 1963 with David Hungerford describing the Philadelphia chromosome, because it was a mutation so big it affected an entire chromosome and we could see them. Of course, we couldn't see the sort of cellular, the gene sequence details for some years later. And in fact, Marie's point about everybody having a different predisposition to different conditions is very true of cancer. I show this not just because it shows the familial nature of cancer. Every one of these blacks filled in squares or circles represents either a man or a woman who's developed cancer within one family and their familial relationships. But the fact that it was published in 1936 from a family that was described in 1911 proves that genetic aspects of cancer, that a familial clustering of cancer has been as fundamental as long as we've known that cancer exists. And of course, we think about that today in terms of particularly breast cancer, the Angelina Jolie effect, the fact that there are clusters of breast cancer in family. But I'm telling you that it applies to everybody. Everybody in this room has a quantitatively different risk of having cancer, something that we've been able to study at the single gene level, as in a BRCA1 single gene, but I can tell you now we're able to take all 22,000 genes and all 6.4 billion nucleotides and add all the independent risks up in principle to give a calculated quantitative risk. And we're learning some really quite remarkable things about how cancer evolves. What's making this possible is this wonderful machine. John Mattick has made the inspired decision to leap into this space before anybody really realized that it was worth doing. It was a very bold decision and tells us something about how we need to make. We need to seize the tempo in science. There's such momentum and such speed that actually timing is everything. It's like a wave. So this machine not only allows us to sequence every one of those 6.4 billion nucleotides in the space of 3 days, just to give you an idea about how amazing that is, it took 8 years and $3 billion, the same amount it took to put a man on the moon to do the one first genome between 1993 and 2001. And we can do it in 3 days now. And what that means is we can do it for a base cost of about 1,700 Australian dollars, 3.4 billion to $1,700. That is like buying a Mac in 2001 for $4,000 and now paying 4 cents for it. So it allows us to apply these technologies beyond these rare families to ask questions of all of us. What is our personal risk of having cancer? What's the pattern in our family? What can we learn from it? And most importantly, what can we do about it? So this is just one paper. I've taken you through 3,000 years, VUM. This is a paper that we published only about 3 weeks ago. And it summarizes the first ever study of the patients that I treat, patients with sarcoma. And it's a global study. You can see all these people come from all parts of the globe. For example, Laura Randall's in Huntsman in Utah. Eva Stratford is in Oslo in Norway and so forth. This is a global study in a rare disease. We needed to do this as a global exercise. And for the first time, we applied these technologies to ask the question, what is the patterns of genetic risk mutations that we see in the germline in patients who develop sarcoma? Now, I should say that sarcomas are cancers of connective tissues like bone and cartilage. And the interesting thing from a genetic point of view is that they're not only associated with some well-described, very rare syndromes, but they affect people at a younger age. So the average age of the patients in our cohort is 20 years younger than the average age of cancer in our community. And that, as Marie was implying, gives a hint that there's a genetic explanation to be found. Now, I'm just going to show you one piece of data, which I think is fascinating. We see lots of multi-cancer families. But we also see families like this, where you have a mother and a father who are unaffected, and then they have 2 kids. Actually, they had 3 kids, because one of the child was lost in the womb and never made it. But these 2 kids developed a total of 3 cancers under the age of 20. And I'm telling you, that's not chance. There's something going on here. But the other thing I'm here to tell you is, it didn't arise from a single gene passing down through the father or the mother, because they would have developed cancer well before they got into their 50s, which is what they are now, perfectly healthy. So what's the explanation? So the explanation lies in this figure here. This figure shows effectively how many people are cancer-free over the course of their lifetime. So, of course, everybody starts out cancer-free, and then they develop a sarcoma or another cancer in our cohort over time. And you can see by the end, everybody's developed cancer, because that's how they got into the cohort. So if you carry no genetic variants that we can identify, you can see that the age of onset of the cancers in this cohort is at roughly, it's 51 years. So younger than average, but not as young as the people who carry this single gene mutation in the gene P53. P53 was identified 6 years before BRCA1 as a cause of very cancer-prone families. Li-Fraumeni syndrome is called. The strongest known syndrome. So it's not surprising that people who carry this mutation in our cohort have an age of cancer onset, which is 32 years of age. That's 18 years younger that they develop cancers. But what was striking was this other group, because now we could measure the effects of multiple genes. We were sequencing 70 genes at once. So we could see people who had more than one mutation, not just the P53 mutation, but something else as well. And for those people, they got the cancer even younger than the P53 mutations. This has never been described before in a quantitative way. It proves that different genes, what Marie referred to as polygenic effects, do account for cancer incidence. And in fact, the fraction in this cohort who had this polygenic effect is twice as large as the group that we currently recognize that carry P53. And what's more important is it starts to explain this situation. Because if polygenic effects are due to different alleles or different variants coming from a mother and a father that combine in the children, then you might have a situation where both kids carry the pair, and so they get cancer, but of course they're separate in each of the parents, and therefore the parents are free from cancer. We begin to understand what we haven't been able to see before. And using the technology, we don't have to be clever or identify these things. We can just ask blindly, what do we find when we look? Because none of these people were identified on the basis of a family history. So what? What does that mean? Does this mean some esoteric mathematical, statistical pleasure to an academic? I'm a clinician. These people are alive. Man is the center of my model system, and there is a duty of care to make a difference. So what I want to talk to you about today is unpublished, and it's the next phase in the story of the Garvan in our attempt to apply this technology, which is 2 years old. So the critical thing is not to get to this point, because you can't cure somebody like this, except with the immunotherapies, which are very exciting potentially in a subset of people. Really, surgery is critical, and for surgery to be effective, you've got to get a cancer when it's like that, a lung cancer that's localized. So what we did was develop a program using a novel technology called whole body magnetic resonance imaging. You see this beautiful, I remember as a doctor seeing MRI come in and the beautiful anatomy that we could see for the first time. It's as spectacular in its own way as all the genomics I've just been mentioning. But it's never been used in a clinical space before. Now the problem with my families is my families get cancer everywhere. For breast cancer families, you can image the breast and you can remove the breast. You can image the bowel for bowel cancer-prone families, and you can remove the bowel. But what do you do when the cancer could turn up anywhere? We didn't have the technology, and that's the reason why the families that I see haven't been introduced into clinical practice. So we had this technology, and we asked, well, if we apply this technology, could we identify cancers early and at a curable stage in our patients? And that turned out to be true. So we've screened 27 people to this point out of the cohort, and we've picked up 3 completely asymptomatic cancers, 2 prostate cancers, high-grade prostate cancers that would have killed these young men. They were under 50 years of age, and a completely asymptomatic sarcoma, the cancer that I treat, in a 52-year-old woman, and all have been treated curatively. So these are 3 lives that have been saved through research that has been conducted and not yet published, and I'm sharing with you today. And it's not just us. You know, you could end up with. We're not a fantasist here. We don't believe in anecdotes and stories. So we're part of a global consortium. This is our little contribution here in the SMOC study. But my colleagues across the world are also doing the same thing. And this shows the aggregate data, which we're going to publish in the next 2 or 3 weeks, months perhaps, where we've screened 462 people who are at high risk of cancer and picked up 1 in 10 people on the baseline scan with an asymptomatic localized cancer. This data, which will be published in 2 to 3 months, will change practice, will change the guidelines that we all adhere to in clinical practice. It's not just about prevention and early detection. Our genomes can tell us things about what drugs will work. And in our cohort, we ended up with 1 in 4 individuals, just down here, who carried a mutation that might have altered our choice of treatment. Should speak more loudly. Yes? Okay, sorry. Well, I did. I treated a patient in the base of this with a drug called a Hedgehog inhibitor because she had a mutation in the Hedgehog pathway, and she's the only patient who's responded. And right now, our plans are all about trying to use this information now to guide therapy. So what we're doing now, today, we've got up to the present, is developing a molecular screening and therapeutics program. We're screening 1,000 people with advanced cancer who don't have any solutions to face. And we're focusing particularly on rare and neglected cancers for which trials don't exist, using a novel signal-seeking trials design. There's a whole lot of technical questions we have to address to make all of this work. And we've opened that. That study is now open. It's been open for 10 days. We've had 3 people enrolled to this point, and we've got 997 spots to go. And we're hoping that we'll be able to offer this potentially life-saving technology to people without them having to pay $5,000 for the privilege of supporting commercial enterprise. And the second thing is, we're taking that cancer risk thing in the germline that we're using genetics for and screening 1,000 people who develop cancer under the age of 40. We don't care what the cancer is. We think if you get cancer under the age of 40, there's something wrong, and there's something that we could find, and we can clearly use that information in a way that might benefit them and their family members. So that SMOC program, Surveillance and Multi-Organ Cancer Syndrome Study, is also open. We have 10 patients who've enrolled, and that's been open for 3 weeks as well. And we will collect another 990 people. And I think by looking through Virchow's microscope at these, what has previously been thought to be an insoluble, outbred, complex problem, we're going to be able to use this information to work out people's individual risk and then tailor the $120 million a year we spend on screening for breast cancer, perhaps to those who are most likely to benefit 10 years younger than we'd ordinarily offer it. And perhaps, the good message here is we'll identify people who are not at risk of having these things and spare them from useless screening and all the anxieties that are associated with it. So this is the vision that we have. You can see we've taken the step, a crucial step across the threshold into returning the information to our patients, and we can see the benefits immediately. And we're trying to systematize it through this program that we're implementing next. And of course, that's something that we can all take part in. Thank you.